Welcome to the presentation on precision coupling with molar amounts of protein or peptide. Topics that we'll cover in this presentation include covalent coupling basics. We're going to look at Avidin beads as an alternative to covalent coupling. And then we're going to look at a workbook for coupling mole amounts of protein per million beads, as well as a workbook for coupling or binding mole amounts of a biotin peptide or protein per million beads. We'll also look at some resources and tools for developing multiplex assays. So let's look at covalent coupling basics first. Whether you're using our microplex beads or magplex beads, both beads are covered with carboxyl groups that, when activated with EDC, will bind to the amines of various molecules. This will allow you to covalently couple a number of types of molecules to the bead surface. These include purified proteins or antigens, antibodies, peptides, some modified carbohydrates, as well as a number of small molecules such as hormones and metabolites and other drugs. You can also couple amine-modified oligos to the beads for the development of genomic assays. In the case of some small molecules, you'll need protein linkers. These protein linkers can be BSA, keyhole limpet hemocyanin, thyroglobulin, or Avidin. Some molecules can also be coupled with small molecule linkers. This includes ADH, MPBH, as well as biotin. In the case of coupling large molecules such as antibodies, these contain a large number of lysines and are relatively easy to couple to the beads in multiple orientations, many of which will work well in an assay. In the case of smaller proteins such as various antigens, these still can have a large number of lysines in them, allowing them to be coupled efficiently and effectively to Luminex beads. In the case of peptides, however, some peptides will have no lysines or a small number of lysines. If they have small number of lysines, such as this peptide here, when folded, the lysines may be in positions that still do not allow efficient coupling to the beads and functioning of that peptide on the bead surface. In this scenario, these peptides can be synthesized with a biotin group at the N-terminus or the C-terminus, and then can be coupled to our biotin or Avidin-coated beads instead. So let's look at Avidin beads as an alternative to covalent coupling. So the Avidin beads take advantage of the fact that Avidin can bind biotin with a dissociation constant of 10 to the minus 15 molar. This is almost as strong as a covalent coupling, like we explained in the covalent coupling chemistry. To take advantage of the Avidin binding capabilities, what you need to do is you need to purchase a bead region for each antigen that's biotin labeled, and then mix that antigen with one of the bead regions, incubate for about 30 minutes, do a few washes, and your beads are now labeled with your protein or peptide of interest. You can then mix these together to make your multiplex mix. Now, whether you're going to be doing covalent coupling or using the Avidin beads, there are some general guidelines that you can follow in building assays with these two approaches. One of these includes the coupling of binding of antibodies or proteins to the beads. In general, you can use microgram amounts of protein per million beads. This allows optimization of an assay sensitivity, especially when coupling antibodies for sandwich capture applications. For small proteins like most peptides, a small molecule linker or protein linker may be needed. These linkers, whether protein linker or small molecule linker, they can also be coupled to the beads at microgram amounts of the protein linker or the small molecule linker per million beads. If binding biotin peptides to Avidin beads, using microgram amounts of the biotin peptide per million beads can also be used. In some situations, however, coupling or binding proteins at mole amounts of protein per million beads is better. This is advantageous when you want to have the signals be representative of the number, not of the number of capture molecules on the bead surface, but the specificity of those interactions, no matter what the size of the protein is that you're binding to each bead. This is exemplified by this data here. In these experiments, 6 antigens were used and they were each coupled to their own bead. The antigens vary in molecular weight from around 20,000 Daltons to 40,000 Daltons. When these are coupled to beads at either 5 micrograms, 2.5 micrograms, or 1.25 microgram per million beads, and incubated with a serum that was specific for antigen D, we see the strong response to antigen D as expected. However, some of the other antigens which should not be reacting to this serum are giving fairly strong positive signals. When we do the same experiment, but couple the beads at picomole amounts, from 20 picomoles to 10 picomoles to 1 picomole per million beads, we see that the signal specific for that antigen is much higher now relative to the background signals that we see on the other antigens. So in order to have this signal more specific to the protein antigen itself, rather than the number of molecules on the bead surface, coupling them at equal amounts of molar amounts of each of those proteins can give you more accurate data for this type of assay. Now, to do calculations for molar amounts of protein per million beads can be tricky. So we have a couple of spreadsheets available that help you with these calculations, and we'll be looking at those spreadsheets today. One spreadsheet is going to calculate for covalent coupling how you can covalently couple molar, nanomolar, or picomolar amounts of protein per million beads, and allows you to do these calculations for scales up to 600 million beads per coupling. We're also going to look at a spreadsheet that allows you to do the same thing with biotin labeled proteins to Avidin beads. It helps you do the calculations of either nanomole or picomole amounts of the protein per million beads. There are also some presentations and files that we have, Excel files, that can do the calculations for doing the microgram couplings of protein per million beads or binding of biotin proteins per million beads. So let's look at the first workbook for the covalent coupling of mole amounts of protein per million beads. So this spreadsheet has a standard format. First of all, there are several tabs. The first tab you'll use is the one called Proteins. And in these tabs, there are several fields. There are green fields and blue fields. And in tabs where calculations are made for pipetting volumes, those will be labeled pink. So you can only enter data into these green fields, and you do not want to enter any information in the blue or pink fields, because that will disrupt the formulas that are used to calculate various amounts and pipetting volumes you'll need for carrying out your experiment. So in this spreadsheet, the first tab you'll go to is the Proteins tab. And in this tab, you can enter the bead region the protein's going to be coupled to, the name of the protein, and then you want to enter its molecular weight. Once this information is added, it's picked up by the second field, the second tab here, the mol/gram calculations, where it will enter the protein's name, the molecular weight you entered, and then convert that to kDa and several other factors as well. Now in this spreadsheet, this green cell here is one that you'll vary, because depending on how many molecules of protein or the molar amount you want to couple per million beads, you'll vary this, because this will be converted into a nanomoles per microliter, which you'll use to determine how much of your protein stock to use in the coupling reactions. So once you've selected a millimolar amount, you'll have this nanomoles per microliter, and then you can come over to one of these other tabs where we're coupling at different scales. And we'll use this first small scale coupling tab as an example. So on this tab, what you'll enter here is you'll enter the bead regions that your protein's assigned to. In this case, it was bead region 18, so we'll enter 18 here. The next thing you're going to enter is this concentration of your magplex bead stock. This can be either 12.5 million beads per ml or 2.5 million beads per ml, depending on which stock you've bought. Information on how to enter that value is listed here on this pop-up. And the other cells have pop-up with regards to how to enter information and what that information is going to tell you when you enter it in the green cells. So let's say you're going to work with a stock of beads that is 12.5 million per mLs. And let's say you want to do some test couplings where you say you want to couple 1.25 million beads per coupling. So I'll enter 1.25 million beads. And then remember that we selected over here on this tab a 0.01 millimolar stock concentration that you want your protein at, which gives us 0.01 nanomoles per microliter. So we want to make sure that this represents the nanomoles per microliter on the other tab. And while this tab will let you do up to 5 different couplings, let's say we're only going to want to do 3 couplings here. So when we enter the 3 for testing 3 different coupling levels of this protein, it tells us now how many beads we're going to need from our stock to carry out the activation of those beads. So when coupling, say, to this bead region 18 3 different protein concentrations, it's going to require that we take 300 microliters out of our stock bead preparation. And then we're going to process that stock bead preparation as described here to activate the carboxyl groups on the bead surface, and then bring them up in a final resuspension volume of 200 microliters in activation buffer. And then the next thing we're going to do is we're going to select the coating level we want. Now, since we are only going to test 3 different coating levels, while the tables down here allow you to do up to 5, we're only going to do 3. So we can delete these tables here by highlighting all the rows, right-clicking with the mouse, and hitting delete. So it gives us our 3 tables that will allow us now to pick the molar coupling level that we want for each bead for each protein titration. So let's say in this example here, instead of 500 picomoles, we want to start at 100. We'll type a 0.1 there. And then for here, let's say we want to do a 10 picomole. So we can add a 0 here. And down here, we want to do, say, 1 picomole coating level. So we can add a 0 and a 1. So now this tells us that in order to take our protein, which is at 0.01 nanomoles per microliter, to do these couplings, we'll need 100 microliters for each of these in 3 different tubes of the activated bead stock. We'll need this much buffer for each of those tubes. And then we're going to need this much protein for each of those tubes. Now, pipetting these volumes might not be comfortable for you. So one thing we can do is we can go back up here and change this concentration to make it more dilute, which means we'll need to add more protein to these reactions. So now we have 125 microliters of protein, 12.5, and 1.25, which should be easier to pipette. But the next thing we need to do is we need to now adjust this amount over on this tab over here. So we can do that by typing over here, adding a 0 here. So that it now makes our nanomoles per microliter equivalent 0.001, which is what our coupling spreadsheet is telling us we need our protein concentration at. So once we've got that settled as far as how we're going to set up the coupling reactions, the next thing we need to do is figure out how much of this protein stock we need to make. And the total volume we need for that is going to be 125 plus 12.5 plus 1. So if we made 150 microliters of this protein preparation, we'd have more than enough to do our 3 coupling reactions. So if we're going to make 150 microliters, you type in that volume here. And then we're going to say if our stock is, let's say, 2 milligrams per ml, this now tells us how we're going to make 150 microliters of our 0.001 nanomole per microliter stock that we need to do the couplings on the next page, where we're going to need 147.67 microliters of activation buffer, 2.3 microliters of our protein stock, and that'll give us our 150 microliters excess of the protein at the concentration we need to carry out these 3 coupling reactions. Once we've got all these calculations done, we're now ready to set up the reactions by pipetting the beads out, doing the washes, following these activation steps, and then arranging these 3 tubes to do the couplings at the 3 different picomole concentrations to determine which one is the optimal one to use for the assay. Once these reactions are assembled, you can then process them as shown in the steps down here. This step is also optional. So in the case of processing these, if you make any changes in this protocol, you can edit this information to reflect those changes. So for example, if you're not going to actually do this blocking step here, you can either delete or strike out this information if you wanted to, and type in that you skipped this step. And skip step 24. Or you can just delete this and then renumber them as needed. And then down here, you can also edit this to represent, say, the final volume you brought your beads up into, which may be 500 microliters or some other volume, and then proceed with testing your beads to confirm coupling. So that's how to use the spreadsheet for doing the covalent coupling. Now, if you're going to scale up, once you've done something, say a small scale testing like this on your beads, and you find out what is the optimum bead coating that you want to use, and you want to scale it up, you'd go through the same process with regards to, if you wanted to entering the information here, changing this as needed, and then say you finally picked on one concentration you want, and let's say that's going to be 10 picomoles. Oops, I want to get rid of this whole table. And we hit delete. And then this tells us that in order to couple 6 million beads, and let's say if we have protein still at this concentration, 0.01 nanomoles, you'll need this much of your bead prep, and you'll take it through this. And then here, this is one of the few cells where you can change the volume for the pipetting volume, so that you now have a total volume of 500 microliters for your reaction. And you're only going to need 60 microliters of the protein prep. So if you're only going to need 60 microliters, you could take this, depending on your pipetting volumes that you feel comfortable with, you can change this to make that protein stock at whatever concentration you need to do your scale up. So that's how you use the spreadsheet for determining how to couple picomole amounts of your protein to your beads. So we have a similar spreadsheet for doing this with Avidin coated beads. Now it's a little simpler than the coupling one, because unlike with the coupling reaction, you don't have to scale the amount of EDC and sulfonyl NHS you need to use. The only thing you need to play with is the total volume that you might want to do your coupling in. But other than that, the format of the spreadsheet is pretty much the same. You'll, again, enter the bead region that you want to couple your protein to in this cell, enter your protein name, and enter its molecular weight. Those values are transferred over to the second tab here, which again is going to give you your millimole and milligram per ml equivalents of your protein, depending on its molecular weight and the millimole concentration you pick here. So let's again go over to here, where again you can enter the bead region that the protein's assigned to, the stock concentration of your Avidin beads, which is generally 2.5 million per ml. And again, let's bump this up to 1,250,000 beads. And again, instead of 5 different titrations, we're going to try 3. And then we can go down here, and since this is again allowing us to do up to 5, we're going to get rid of the 2 tables that we don't need. Hit delete. Then again, we have our bead region, the stock concentration of our Avidin beads. For the testing of 3 different concentrations to bind to the beads, we're going to do 1,250,000 per bead region. And then we're going to, let's say, bump this over to the 0 here, see if it works for this particular protein. And so this is telling us that we're going to need 1.5 mLs of our bead stock to do these 3 different couplings at 3 different concentrations. Unlike with the coupling chemistry, all we have to do here is wash the beads in some suitable buffer, which can be PBS, TBN, or several other buffers as the pop-up indicates here. Take it through the various washing steps. Then again, resuspend in some final volume. Now again, most pink cells you cannot edit, but this is the exception where if needed, you can edit this one, depending on the final volumes you want to do your reactions in. So once you've done the washes, you can then determine, okay, well, what reaction volume do I want to do each of these reactions in? And let's say we want to bump this up to 1,000 per reaction. And then this we can make this smaller. So let's make this 300 microliters. And then the next thing we're going to have to do is figure out how to, actually we need another 0 here, it's supposed to be 1,000. Then let's say we're going to drop this back again to the 100, and then we're going to do the 10 picomoles like we did for the covalent coupling. Then we're going to do the 0.01 picomole like we did, or the 1 picomole for the covalent coupling. So once you've done all these calculations here and added these values in, this again tells you how many microliters of the protein you need, which again, 150 microliters would be more than enough. So you can drop this down to 150 microliters, and say your protein is still 2 milligrams per ml. So this now, oops, we have to adjust this because it's 0.01 nanomoles per microliter. We go up here, 0.001 nanomoles per microliter. That's equivalent to 0.001 millimolar. So now this tells us how to make, again, how to make our 150 microliters of stock at this concentration, which for this protein turns out to be this milligrams per ml. And then we can follow this protocol for doing the washes, resuspending, and then splitting that resuspension up into 3 different reactions to do our 3 different bindings for 30 minutes with several washes and a final resuspension. And again, if you make any changes to these steps down here, you can edit these sections as needed to include any changes you did with washing volumes, number of times, and with regards to the final resuspension volume, which volume you finally resuspend the beads in. This tab here does pretty much the same thing. The only difference is that if you want to pick automatically a different fold dilution to use for your different protein titrations, you can just pick a different number here if you wanted to try a different range of titrations of, say, 4-fold versus, say, a 2-fold first concentration. So once you've got all your parameters set, and you have your calculations for how to make your protein stock to do the binding with, you're now ready to mix everything together and then follow your procedure and have your biotin coated beads within an hour and ready to test them further to develop your assay. So once you've got your beads coupled covalently, or if they're bound to Avidin beads, the next thing you want to do is you want to determine the bead concentration. Because during the process of doing these couplings or bindings, you could start with, say, 1.5 million beads in a reaction, but you will not recover all of them. So it's recommended that you get either a hemocitometer and a microscope or some type of cell counter to determine the concentration of your beads in each preparation, because you could have significant or maybe very little bead loss. There's a couple of spreadsheets that are designed to help you with these calculations, because once you've done those couplings and bindings, if you're coupling, say, 10, 20 different bead regions, you want to make sure that when you start using them downstream in any reactions and assays, that you have the right amount of beads added to each reaction. And one of the best ways to ensure that is that once you've done your couplings or your bindings, you determine your bead concentration, and then you want to dilute your beads to some fixed concentration. That can be anywhere from 500,000 beads per ml to 1 million beads per ml. In order to do that, the spreadsheet has 2 tabs that help you with those calculations. And we will not go over those spreadsheets here, but if you need to use these spreadsheets to normalize your bead concentrations for building your multiplex assays, you can contact your FAS and they'll give you assistance with how to set this up to start making your multiplex bead mixes. Once you've done that, we also have a calculator that helps you with those multiplex bead mixes, because mixing all these beads together can be complicated, because depending on the plex of the assay, there will become a point where you may even have to concentrate your bead prep in order to make sure that you're delivering the right amount of beads to each reaction. So we have this bead mix calculator, which is available online for doing that. If you need assistance with this, you can also contact your FAS for support on how to build your multiplex bead mix using this calculator. So additional resources for building assays can be found in the XMAP cookbook. Cookbook is a collection of easy-to-follow protocols that also has lists of reagents and equipment that you need to build your Luminex XMAP assay. This can be downloaded from the link here. If you want a copy, you can get one from your business manager or FAS. Well, that concludes this presentation on precision coupling of molar amounts of protein or peptide. We'll have some other presentations available as well, which includes doing calculations for coupling or binding microgram amounts of proteins or peptides to your beads, as well as the tools for doing the bead concentration calculations and making your multiplex bead mix with those two calculators I just showed you. So thank you for listening to this seminar. If you need any additional assistance, please reach out to your business manager or FAS. Thank you and have a good day. Okay, so that should be it for now, Scott. I don't know if you want to go back